Hello friends, good morning to all of you. Today I am again back with Plant Growth and Development, first part of 11th standard. In this session, we are going to discuss growth, plant growth, growth is measurable, phases of growth and growth rate. So let us start the session with growth. Growth is one of the most fundamental and conspicuous characteristic feature of living being. It is applied to the whole series of changes which an organism goes through during its life cycle. Thus, growth is defined as a permanent and irreversible change in size of the cell, organ or whole organism which is accompanied by an increase in dry weight. Growth takes place due to metabolic process with the expense of energy. For example, expansion of leaf is growth. Here in the given figure, you may observe the germination and seedling development in beans, thereby growth. Now about the plant growth. In case of plant, the growth is unlimited. So generally it is indeterminate. Growth occurs continuously throughout the life by the activity of meristematic tissues. The new cells are continuously produced by cell division in a meristem. Such kind of growth where new cells are continuously added to the plant body with the help of meristem is also called as open form of growth. In plant, both primary as well as secondary growth takes place. So we will see what is the difference between primary and secondary growth. In case of plant, different types of meristematic tissues like root apical meristem, shoot apical meristems are present, and these meristems are responsible for primary growth of plant. The root apex and shoot apex meristem add new cells to cause linear growth parallel to the long axis of plant body. It means it results in elongation of plant along their axis. But in case of dicotyledonous and gymnospermic plant, there are lateral meristem, vascular cambium and core cambium, and this appears later on, and this helps in increasing the girth. This is called as secondary growth. In the given figure, it is shown about the diagrammatic representation of the sites of shoot apical meristem and root apical meristem and also vascular cambium. Here the direction of growth is shown with the help of arrows. The figure clearly shows that root and shoot meristems helps in elongation, whereas vascular cambium helps in girth of the plant. Now the next topic is about the fact that plant growth or the growth is measurable, means it can be measured by different parameters. So the different parameters used to measure growth are increase in length or girth, increase in fresh or dry weight, increase in area or volume, increase in number of cells produced. So for measuring the growth of plant or plant organs, we have to choose any kind of parameter. For example, one single maize root apical meristem can give rise to more than 17,500 new cells in an hour, whereas cells in a watermelon may increase in size up to 350,000 times. Thus, in maize, the growth is measured by increase in cell number, whereas in watermelon, it is measured by increase in size of the cell. Growth of pollen tube is measured in terms of its length. In dorsiventral leaf, it is measured by increase in the surface area of the leaves. So, these are the different parameters by which growth can be measured. Now, the next topic is about different phases of growth. There are three different phases of growth. They are meristematic phase, phase of elongation and phase of maturation. So here we are going to discuss one by one. First is about meristematic phase. Cell is metabolically very active at the time of meristematic phase. Cell division takes place in the root apex and shoot apex. These regions are also called meristematic zone, where cells are in a state of continuous division. The cells in this region are rich in protoplasm and possesses large and conspicuous nucleus. The cell wall are also thin, made up of cellulose, and it consists of large number of plasmodesmatal connections. Now the next phase of growth is the phase of elongation. Cell division is followed by cell enlargement before maturation takes place. The cell just next away from the tip to the meristematic zone represent the phase of elongation. In this region, the cells are having large vacuole, large cell, and there is new cell wall deposition. The new cell wall deposition is synthesized to cope up with the enlargement. Now the last phase of growth is phase of maturation. The region of maturation is more proximal to the phase of elongation, and the cells of this zone attain their maximal size in terms of wall thickening as well as protoplasmic modifications. Thus, in this phase, cell differentiate. For example, the meristematic cell of leaf epidermis differentiate to form guard cell, trichomes, glands or epidermal cells. The differentiation and maturation of various kind of cells depend on the number of internal, external, nutritional, genetic and hormonal factors. The given figure indicates about the zones of elongation in young root. Now the next topic is about growth rate. Growth of an organism or part of organism is expressed in form of growth rate. It is defined as increase of growth per unit time. The growth rate of an organism may be expressed mathematically and it may be expressed arithmetically or geometrically. We will discuss here both separately. First, it is about arithmetic growth. In arithmetic growth, following mitotic division or mitotic cell division, only one daughter cell continues to divide while the other differentiate and matures. It is seen in root apex and shoot apex. In this case, single meristematic cell divides mitotically to form two daughter cells. Then one of these daughter cells differentiate and become a permanent cell, whereas the other one remains meristematic and again divides by mitotic division. And this process continues and the new cells are constantly added to the body, resulting in the constant elongation growth. If the length of the growing organ or the organism is plotted on a graph against time, then a linear curve is obtained. Mathematically, it is expressed as LT is equal to L0 plus RT, where LT is the length at time t and L0 is the length at time 0 and R is the growth rate divided by elongation per unit time. Now it is about geometrical growth. In geometrical or geometric growth, after mitotic division, both daughter cells continue to divide mitotically and it will continue to do so. Here in most systems, the initial growth is slow and it is called lag phase, but it increases rapidly, called as exponential phase. But when the nutrient supply is limited, the growth slows down, leading to a stationary phase. Thus, if we plot the parameter of growth against time t, then we get a typical sigmoid or S curve. Mathematically, the exponential growth can be expressed as W1 is equal to W0 ERT, where W1 is the final size, it may be the final weight, height, number, etc. W0 is the initial size at the beginning of the period, and R is the growth rate, T is the time of growth, and E is equal to base of natural logarithm. Now you can see in this figure that about the arithmetic and geometric growth. You can observe here that in arithmetic growth only one cell is dividing, whereas in geometric growth both cells are dividing. The given figure shows about the stages during the embryo development, showing geometric and arithmetic phases. This is a graph showing constant linear growth, a plot of length L against time t. This is the graph showing sigmoid growth curve. Here you can observe lag phase, exponential phase and a stationary phase. Now the last topic of today's session is about growth rate. Growth rate is the comparison between the growth of living system. It is of two types, that is absolute growth rate and relative growth rate. If measurement and comparison of total growth per unit time is taken, then it is called absolute growth. And when the growth of given system per unit time is expressed on a common parameter, then it is called as relative growth rate. The absolute and relative growth rate can be explained using the growth in size of two leaves. This figure indicates that the absolute growth rate is same in both the leaves, but the relative growth rate is different. The relative growth rate of leaf B is more as compared to that of A. So this is all about today's session. Thank you. For any kind of doubts, please write your question in comment section.